Welcome, guys. In this lecture, we shall discuss the concepts of morulation, blastulation, and gastrulation. Once the zygote gets formed, in case of humans, it undergoes cleavage. By continuous cleavage, this zygote gets changed into the solid ball of blastomers, which we call as the morula. Morula literally means the mulberry fruit, and the morula is composed of a ball of blastomers. This is the diagram of the morula. As you can see, it's a solid ball of blastomers. Blastomers are the cells which are formed as a result of cleavage, and the morula is composed of 16 to 32 blastomers, and it has an inner cell mass. This is the inner cell mass. These shaded blastomers present the inner cell mass, and this outer layer of cells, this outer clear layer of cells surrounds this inner cell mass. Then is present the zona pellucida. If you have understood the structure of the ovum, you might have seen that this zona pellucida is one of the layers which covers the this ovum, so it prevents the sticking of morula at any improper place. As this morula reaches into the this uterus and gets converted into the blastula, then it gets implanted in the uterus after the degeneration of this zona pellucida. So this zona pellucida prevents the sticking or implantation of the embryo at any improper place. If the embryo gets implanted at any improper place, that it doesn't get implanted at its actual place of implantation, which is the uterus. This condition where the embryo does not get implanted in the uterus but gets implanted somewhere else, like in the fallopian tubes, is known as the ectopic pregnancy. The morula is formed about the morula reaches the uterus about 4 to 6 days after fertilization. Then the morula changes into the second stage, which we call as the blastula. This is the blastula. Now what happens during the blastulation? Once this morula reaches into the uterus 4 to 6 days after fertilization, this now loses the zona pellucida, and this outer layer of cells is now known as the trophoblast, or it is known as the trophoectoderm. This is the trophoectoderm. Now through the trophoectoderm, from the endometrium, the fluid is absorbed and it creates a hydrostatic pressure inside this structure, which leads to the pushing of this inner cell mass towards one pole, and this embryonal knob of cells gets pushed towards a single pole, which was earlier known as the animal pole. It is now known as embryonic pole. The opposite pole, this very pole where a cavity is formed, is known as embryonic pole, and the cavity formed over here, this cavity is known as the blastocele, and it contains the fluid which has been absorbed through these trophoectodermal cells or through these trophoblast cells. Furthermore, one more thing is that the embryo gets implanted at this very stage, that's at the blastula stage in the uterus. In mammalian embryos, this blastula stage is also known as blastodermic vesicle or the blastocyst. So after that, now the next stage is the implantation. Implantation means that this structure over here, that is the blastula, it gets implanted in the endometrium of the uterus, and this is what we call as implantation. Implantation occurs by the activities of both the blastula as well as the endometrium. Some of the trophoectodermal cells, these are the trophoectodermal cells surrounding the blastula, they secrete some lytic enzymes which degenerate a part of the endometrium over here and make a pit-like structure, this pit-like structure in which this blastula gets embedded, and the blastula then grows the villi into the endometrium, whereby it absorbs the nourishment from the endometrium and grows. Later on, it establishes a firm connection with the endometrium so that it derives all of its nourishment from the endometrium. This is, after zooming this diagram, you see this is the blastula stage and how it gets implanted in the uterus. The implantation of the blastocyst in the endometrium occurs roughly at D7. This is the manner, you see, these are the fallopian tubes. This is the manner in which this morula moves towards the uterus, then gets converted into the blastula and gets implanted in the endometrium of the uterus. Now, after that, now we shall focus on this set of diagrams whereby we understand the mechanism of the gastrulation. After the blastulation is over, now starts the process of gastrulation. Now, first of all, how we understand what is gastrulation? Basically, in the gastrulation, the embryonic cell mass, this embryonic knob, it begins the movement of cells and establishes three germ layers, that's endoderm, mesoderm, and ectoderm. This movement of cells, which results in the establishment of these three different germ layers, is known as the morphogenetic movement of the cells. Now, how it happens? This is the embryonal knob. It contains the inner cell mass. This is the trophoectoderm. On the lower margin of the embryonal knob, some cells begin to proliferate, as have been shown by this lower layer of cells which are not shaded, and this forms the first layer of cells which is known as the endoderm. Now, what happens after the endoderm begins to proliferate over here? It doesn't remain confined to this area, rather it gradually begins its invasion downwards. This is how the endoderm gradually invades the whole of the blastocele, and it lines the blastocele on its inner side. Earlier, the blastocele was only lined by a trophoectoderm. Now, once the endodermal cells proliferate, they also line the trophoectoderm on the inner side. And now this cavity is no more known as blastocele, it is known as the primary yolk sac. In many of the competitive examinations like the NEET, you are asked that primary yolk sac stores. The options are it stores the yolk, it stores the fluid, it stores both, or it stores none. The simple answer is that it stores the fluid, as you can simply understand that basically it was blastocele. Now, once the blastocele gets lined on its inner side by the endodermal cells, it forms the primary yolk sac. So it is very simple that it doesn't contain any yolk, it simply contains the matter which was earlier contained in the blastocele, and that's the fluid. Now, after that, what happens later on? Once the endoderm is formed, what happens? This layer of cells, now it forms two sublayers. That's the upper layer of the cells, which is known as epiblast, and the lower layer of cells, that's known as the hypoblast. This hypoblast layer of the cells is the layer of cells which has formed the endoderm. Now, from the epiblast, two more cell layers will be formed, that's the mesoderm and the ectoderm. Now, what happens? This layer of cells becomes structured, and over here the cells which earlier connected the trophoblast with this cell mass, which were known as the cells of Ruber, they break away and they generate a cavity which is known as amniotic cavity. The amniotic cavity in its roof is lined by a category of cells which we call as aminogenic cells. This outer layer of cells is the trophoectoderm, and this layer of cells, these cells, it is the aminogenic cell layer. And this cavity formed over here is the amniotic cavity, and this cavity over here is now the primary yolk sac. Now, let's understand how the mesoderm and ectoderms are formed. Now, this portion, if we discuss about this portion of structured cell layer, it is known as the embryonic disc. This is the magnified diagram of the embryonic disc. The embryonic disc is divided into three parts. That's the cephalic margin of the embryonic disc. This anterior margin of the embryonic disc is known as cephalic margin. Then there's embryonic disc proper. Then there is the caudal margin of the embryonic disc. This is the endoderm, or this is the hypoblast. This layer is the epiblast. This hypoblast layer forms the endoderm. Now what happens at the caudal end, or at the caudal margin of the embryonic disc, there begins proliferation of cells, as has been shown by this immigrating mesodermal cell layer. There begins the proliferation of cell layer which forms the mesoderm. Now you can understand it in this way, that the mesoderm begins to proliferate and it invades, and how all other these sequences happen is like this, that now the mesoderm, it penetrates in between the trophoectoderm and the aminogenic cells on the amniotic cavity side, and it traverses, or it invades in between the trophoectoderm and the endodermal cells on the what we call as secondary on the primary yolk sac side. So this layer of cells, which has been shown by this dotted area, is now known as extra embryonic mesoderm. Now after that, what will happen? This is the primary yolk sac, this amniotic cavity. Now after the mesoderm is formed, the remaining part of the epiblast, after the formation of mesoderm, forms the ectoderm. This is the ectodermal part. Now after that, what will happen? Now the mesoderm will split. It will split into two layers. This is the visceral layer of the mesoderm, which surrounds this area, and this layer is the parietal layer of the mesoderm. And once the mesoderm splits into the visceral layer of mesoderm and parietal layer of mesoderm, the cavity formed between these two layers of the mesoderm is known as the coelom, and this coelom formed by the splitting of mesoderm is known as schizocele or schizocoelom. One point we must understand over here, that this is the manner in which the three germ layers have got formed. But we should remember one more point over here, that's this endoderm which was earlier formed, it forms the digestive tube. So this is how the gastrulation occurs, and after that the three germ layers formed, that's the ectoderm, endoderm and mesoderm. They establish all the organs which are present in the human fetus, and that we shall be discussing in a separate lecture under organogenesis.